that you?
No. Yeah, it's Tanya. All right. It's one question, but it'll get our attendance. I don't have tonsils anymore, but when I had tonsils, I got it all the time.
I had my first tonsils last year at 17 years old.
They tend to ask us for our emails.
Yeah.
Yeah, just put your name so I know who you are. Yeah.
I had
Something to do with your name so I know who you are. Sorry, I forgot to ask for the email.
I had tonsils the first time at 18, what was that, 18 or 17, I don't know, recently.
Yeah, I didn't get so I got my tonsils out when I was 37.
For you.
40. I'm 41 this year. Yeah. Man, millennials, I'm telling you, millennials look younger right now.
So graceful.
Thanks. Well, it says something because I feel like smoke, so it shouldn't be helping. It's cigarettes, yes.
Ooh.
Yeah. Don't ever smoke cigarettes. If you haven't smoked cigarettes and you don't smoke cigarettes, don't.
Don't
Because they
worry.
are actually like the addiction is real, I'm telling you.
My dad.
Just relax.
My dad dropped lucky that day.
There's a point to push it. Right.
My dad dropped it in my mouth. Never smoked that. Never.
Yeah, that'll probably do it too. Yeah. So my mom smoked while I was growing up, and I was like always like that kid that was like telling her how it was bad, you need to quit, like this is gross and all. She told them her nasties because people told her it was nasty and all this. But I don't know, man. When you get in your rebellious years,
No.
things can happen.
Okay, I'm a.
I didn't start smoking until I was almost 30. So
I think the worst thing I've ever done. I was 17. I was working at Subway, and I took a hit of somebody's weed, like, but I needed it.
sometimes
Rough.
sometimes you need it. Sometimes you need it.
And I'm sorry, I'd rather do a herb than that literally burgers your
Well, tobacco is an herb.
yeah. It's just not very pure anymore.
As it turns out, and I am not condoning smoking. I just want to be clear on
Don't
this.
smoke.
Don't do it. But as it turns out, nicotine has an effect on cancer that actually inhibits it. Like it can prevent cancer, nicotine can. So you know how we hear about people like you smoke and it leads to lung cancer? Most of the time, in almost all the cases, people who quit smoking. It comes after they quit smoking. The nicotine, like, I hate to say it, but that is. Don't smoke, just stay away from it, and you'll be better off.
But I also like severe bronchitis, you know. My lung.
Plus emphysema and COPD, like those things are really hard to don't smoke. Yeah.
But my question would be, if it's just cigarettes, like are we talking tobacco?
Yeah, you know.
For nicotine,
My mom and dad used to smoke, but they both quit when they
so
found out
what
they were
nicotine
pregnant
does is
with my brother.
your ATC.
But then, my dad dips, and I used to lay on her chest all day.
Yeah, so if I know how when you're
And I can, to this day, still feel
your DNA,
back with them.
and it can cause mutations from that can lead to
But it also,
the same thing. And then you can have it can cause them to not get it.
So it's overall.
That's DNA.
And it's also.
Yeah. No carcinogen, it probably don't cause cancer.
But I was just wondering, like, it's just.
I mean, I know you were an adult. I didn't know, like.
But there are other
older.
tobacco.
Everybody.
And I feel like those are two, and that those guys, other chemicals in tobacco, those are.
My mom is a Dax.
Guys, I've
You
been
were
born
1980.
and putting one of those things, you do not want to.
I was
Don't-
raised basically as a.
Don't follow in my, don't follow me. Do not do as I do, okay? All right, are we ready?
But you can't quit.
Yes, well, you can. It is I'm telling you, if you haven't smoked, you don't know, man. It is hard.
You can go to Vegas and
Then you're addicted to
I have a
vaping.
favorite vape by Nature Place.
The same.
I was like, I'll wake up and vape and go sleep.
Yeah, that's what my husband does. He sleeps with his vape, like literally like in his hand.
Like, I'll look over and hit my vape.
So I try refreshing it or I get out of it.
Okay, for free.
All right, cool. So the general consensus on this one was blue is the most favorite, then green. Who cares? So we got we got our data for the morning. All right, so we're going to be lecturing over chapter 11 today. Chapter 11, we're going to be talking about physical and chemical control of microbes today. So some of the terms, some of which we've already kind of introduced ourselves to, but some of the terms we want to be aware of during this. Oh, whoops. Sterilization, disinfection, antisepsis, and decontamination. So sterilization means complete removal of all microbes, okay? So that means all endospores, all viruses, everything is gone. There should be no evidence of microbes there at all. So that's sterilization. Everything's dead. Disinfection is destroying most microbial life. Generally speaking, disinfectants like Lysol and things like that, are disinfectants will not be getting rid of our endospores. Generally speaking, it's the endospores that are the hardiest ones that will survive the disinfection. Okay, so disinfection does not mean getting rid of everything. It means getting rid of most of the stuff, all right? Antisepsis is like disinfection, but on animate surfaces, like on body. So whenever you're prepping to start an IV or, you're getting ready to do a finger stick and you're using an alcohol wipe, or if you're just using hand sanitizer, that's sanitization of the skin, that's still considered antisepsis. Has to deal with a living surface, that's antisepsis. We also call it degermination, but I'm never going to test you on that. Just FYI, I'm always going to call it antisepsis. So an antiseptic. You guys have heard this word before. Decontamination, we can also call this sanitization, is the mechanical removal of microbes from, it could be an animate surface or an inanimate surface, doesn't matter. But if you're mechanically removing microbes, we call this decontamination. So this could be washing your hands. The suds are like actually physically removing the microbes from your skin. It's not that the chemicals in the soap is necessarily killing the microbes, but that the sudsiness of it would lead to the removal of the microbes physically. So that's decontamination. Okay, so you're physically removing these microbes or excluding them somehow, that's decontamination. So like filtration, just physically removing microbes, so that's decontamination. All right, so this is a great chart for your study purposes whenever you guys are going through organizing some of the stuff that we're going to be learning about today in your own study purposes. So we're going to go through all these categories kind of one by one, but it does break it down as to whether these processes are capable of sterilizing or just disinfecting or antisepsis and things like that, okay? So what I want to be clear is it will sterilize if it's used appropriately. So plenty of these things have the ability to sterilize, but we aren't using them appropriately. And so they're not actually sterilizing, okay? So keep that in mind whenever you guys are studying. Endospores are considered the most resilient of the microbes by far. We know that they are developed specifically to be hardy and to be resistant. So destroying endospores is usually the goal of sterilization, because if you can destroy endospores, you're almost certainly destroying everything else. Okay, so if you can get to that level of destroying endospores, you're good to go, man. So the goal of sterilization to destroy all life forms is typically to go that extra step of destroying the endospores. Because like we said, if it kills endospores, it'll kill everything else. So disinfection and antisepsis, they're going to act on organisms that are typically less hardy than our endospores. So again, anything where you have treated it to remove all life forms, even the endospores, that object will be called sterile afterwards. That's what sterilization is about, removing all of the life forms. There are things, of course, that we use in our lives where we would need it to be sterile and not just disinfected. It's fine to disinfect your tabletops, to disinfect your, forks and knives and spoons and things like that at home, but you don't need to sterilize them really. Most of the time with our healthy enough immune systems, sterilizing everything isn't actually going to make any difference. So we don't always need to sterilize everything, but there are some things that absolutely do need to be sterilized, like surgical instruments and syringes. And certain packaged foods will also be sterilized. That helps with the longevity and preservation and things like that. So disinfectants are usually used on inanimate objects. Again, we're not killing everything. Usually endospores are left alive, right? Disinfection process also typically will remove harmful products, not just the microorganisms, but their toxins that would be associated with them. Not just removing, Clostridium botulinum, but also the botulism toxin. Antisepsis, again, this is whenever we are talking about growth of, preventing growth of microbes on living things. So sepsis is a term that refers to microbes growing in tissues where they shouldn't be. That's sepsis. And when we talk about septicemia, that's a term that's referring to microbes that are flourishing in the bloodstream, right? So sepsis, septicemia, microbes growing in place in our tissues. Asepsis is anything that prevents the infectious agents or any microorganisms from entering tissues or whatever environment we're working with. Because we learned about aseptic technique. Aseptic technique in our lab and aseptic technique in hospitals and things like that, they all have the same purpose: to prevent contamination. That's the goal, to prevent contamination. It's not to exclude only true pathogens. It's not to make sure things are maintained sterile necessarily. You want to keep things as sterile as possible, but the real goal for aseptic technique is to prevent contamination. So the techniques that we use might be sterile methods to exclude all microbes, but it also generally the whole goal of it is to prevent contamination. So antisepsis is the application of any agents called antiseptics that will be exposed to living surfaces. It could be open wounds, it could be just intact tissue or whatever. All right, so we already learned from our Kirby-Bauer assay from day 3 on Kirby-Bauer about the cidal versus the static. So anything that's called cidal means that we're killing something. So our big term, we learned about this with bacteria. So it's bactericidal or bacteriostatic, right? But the big term for this is going to be germicidal or microbicidal versus germostatic and microbistatic. Okay, that's all microbes. But bactericide, bacteria, fungicide, fungi, etc. If it's called a sporicide, it kills endospores. Okay. Static agents, these are things that inhibit the growth, but it doesn't kill the microbes. All right, decontamination, again, also known as sanitization, mechanical removal of the microbes. So this is often used, the best example we have is using soap. Literally, the suds are going to prevent them from sticking to the surface of your own cells, so we can wash them away physically. Yes.
I'm sorry, will you please go back to the previous slide real quick, so I can just, thank you, that's all I needed.
All right. So again, decontamination, the mechanical removal of the microbes or any other debris. So if you're washing your hands and it's making the dirt go off of your hands, okay, that's decontamination. Okay. All right, so the reason for microbial control on devices, we have the critical, the semi-critical, and the non-critical devices. It's a critical device if it comes into contact with sterile tissues. So if we're cutting into your body, there shouldn't be microbes just hanging out in you're actually in the layers of your skin, the deeper layers of your skin. There's not microbes in that. On the surface, sure, but there shouldn't be at deeper levels. In your blood's completely sterile. We don't want to be introducing microbes into places where they don't belong. We want to keep them how they are. And so any time that we're opening up somebody and it's potentially exposing them to microbes that wouldn't be inside of their body, that is a critical medical device that would be involved in that, right? Then we have semi-critical medical devices. These are devices that come into contact with our mucosal membrane. So there's more likelihood of microbes being able to get in through your mucosa than there is through intact skin, for example. Those are our semi-critical devices. So this would be like respiratory equipment and things like that. Then we have non-critical medical devices, things that don't touch the patient or are only exposed to intact skin. So this would be door handles, sheets, the bed itself, anything like that. All right, cellular targets for our chemical agents. Most of our chemical agents and our physical agents actually will be targeting these 4 aspects of our microbes, either the cell wall, the cytoplasmic membrane, the cellular synthetic processes. This could be making protein or making DNA or RNA. Okay. So things that affect the cell wall, you can either block the cell wall from being made, so blocking cell wall synthesis. You can digest the cell wall. You could start breaking down just the surface of the cell wall, so leading to a disruption of the integrity of it. So anything that has a damaged cell wall is more likely to be more easily lysed. And because we've already talked about our gram positives, they have a thick cell wall. They really depend on that thick cell wall. They don't have any other thing to help out with the surface of their cell. They have their inner membrane, everybody has that, but that's just not enough. So they also need that cell wall there. Gram negatives, they have the thin cell wall, but they also have the outer membrane. So they can take some damage to their cell wall and still truck on pretty well. But our gram positives, they need that thick cell wall for the environments that they live in. So anytime that you're affecting a cell wall, a lot of times our gram positives are more significantly affected by that. Detergents and alcohols can disrupt the cell wall. You can also affect the membrane. So if you're affecting a cell membrane, remember your membrane is selectively permeable for a reason. So you can use agents that will lead to loss of that selective permeability, which would lead to loss of the molecules inside of the cell. That would lead to the potential death of these organisms, as well as allowing entry of other chemicals to get in. A lot of the times things that affect the cell membrane aren't going to kill the organisms, but they'll set them up for other chemicals to be able to get inside, and those can then kill. Surfactants are an example of things that can affect the cell membrane. 1, 2, 3, 4. Oh, geez. Surfactants will affect cell membrane because they are polar, they're amphipathic. Okay. They have a polar region and they have a non-polar region. And so surfactants often look like this. They either have a charge, it's either negative or positive charge on a head, and then they have a long hydrophobic tail. Look familiar? That's exactly how they work. They're going to squeeze in between your phospholipids in your membrane and cause them to be disrupted. All right. So it looks like this. There's your phospholipid bilayer, and our surfactants, which have a similar property. They have a charged head, negative or positive. And then they have that hydrophobic tail. And that mimics the shape of our phospholipids. And so they'll squeeze in there, but that disrupts the integrity of the membrane. And now things can get inside of the cell through the disruption. Also causes things to leak out of the cell as well. So things that affect protein and nucleic acid synthesis. If we're affecting nucleic acid synthesis, we have molecules that can like mimic your A, T, C, or G and can replace them and cause inhibition that way. We can also inhibit the actual transcription and translation processes. So we can also inhibit the ribosome from being able to function properly too. We already know about our protein state, the native versus denaturation. If you're normally folded up, something like this, usually that stuff that's on the inside is hydrophobic. And stuff that's on the outside is the hydrophilic, because it's exposed to the water. And that if you denature these things, that disrupts that whole, how the protein is folded, therefore how it functions, how it interacts with its environment, and all that sort of stuff. And that can significantly inhibit a microbe being able to function. Because obviously, if you introduce a chemical that denatures DNA polymerase, that microbe can no longer replicate its DNA and therefore make copies of itself. Can't do it anymore. It's be inhibiting its growth by doing that. So denaturation is disrupting those proteins so they don't function anymore. Here are some things that can disrupt our proteins. Moist heat. Heat is very effective at disrupting our proteins. Moist heat, the adding of the water helps with the denaturation process. So interacting with all those R groups on our amino acids and then leading to them being destabilized. So when you think of cooking an egg white, when you're boiling it, you know, once you have boiled that egg, is there any going back to the original egg? No, right? So you've denatured those proteins, permanent. A lot of the times that will be the case. So they can't revert back to how it was. So we also have organic solvents that can have the same effect. Phenolics are a great example of that. So that could be phenol itself, which is, you know, a benzene group and an OH group off the top of it, Or it could, that's also called carbolic acid, which is what Lister used for his operating rooms. But anything that's derived from phenol is called a phenolic. We're getting into that today. Metallic ions can also bind to the active site to competitive inhibition, right? So certain metallic ions can lead to competitive inhibition. That would lead to destroying the function of the protein as well. Is this not recording? Just something else. I'm sharing this right on the Zoom, right? This is sharing. Yeah, we're recording. Okay, I just want to be sure because it popped up with the thing saying, okay, never mind. All right, so we have your normal protein that would have the native state right here. And these are the 3 different ways that you could affect it. Normal protein, where your substrate binds and how the enzyme is going to be interacting with it. If you denature it, it can't interact with the substrate anymore. If you change its shape somehow, that can't interact anymore. And you can also block the actual active site with agents like metallic ions, competitive inhibition. Okay. Methods of physical control. Let's jump into these guys. So these are methods of applying something physical to an item or a surface or whatever it is. And then that physical aspect of how what you're applying to that organism will lead to sterilization or sanitization. Okay. So heat is obviously the most commonly used one. If you ever worked in a hospital or a vet office or a dental office, then you've probably heard of an autoclave. And so an autoclave is the best example that we have of something that uses heat and moisture in order to sterilize. So autoclaves are capable of sterilization, so complete and total removal of any living organisms. Other methods that are used commonly would be radiation, filtration, possibly ultrasonic waves. We're not going to be talking about that in this class. And then cold is another way to prevent microbial growth. All right, so heat. Hot water, boiling water, and steam count as our moist heat methods. Okay, temperatures for sterilization with moist heat or for basically removing microbes in general, you range from 60 degrees Celsius up to 135 degrees Celsius. Remember, boiling is 100 degrees Celsius. Okay. The temperature of the steam can be regulated by adjusting pressure in a closed container, which is what an autoclave does. We add pressure as well as the temperature in the water, and the combination of those 3 things is what allows for sterilization. So this allows us to operate at lower temperatures and shorter exposure times than what we would see with dry heat. We're going to show you in a minute the difference between dry heat versus moist heat and the effectiveness involved there. So the idea is that by adding in the moisture, you can coagulate the proteins more effectively. So the heat plus the moisture are going to coagulate this protein so they can't be as active. So dry heat, you can also use dry heat for controlling microbes. This ranges from 160 degrees Celsius up to several thousands of degrees Celsius. And most of the time we're operating in the several thousand range, not always. This will dehydrate the cell or completely oxidize them, leading to ash. So that's what we see with incineration, right? We're completely oxidizing the cell and its components, and then that leaves us with ash in the end. So that's oxidizing that's happening there. So here's a comparison between the effectiveness of these guys. With moist heat at 121 degrees Celsius for moist heat, 121 degrees Celsius, you would expose it for 15 minutes and you get complete sterilization, right? Anything less than that, you still have decontamination, right? You still are removing microbes. Disinfection is the proper word for it. You'll still be disinfecting, but you would have endospores if you don't go for that full time at that temperature, right? So that's what I'm saying. Using moist heat doesn't just mean you apply some heat and some moisture and immediately the thing is sterilized, right? You have to follow the rules. And if you're following the rules appropriately, you can sterilize. Otherwise, you're just disinfecting. Okay, so in order to sterilize, moist heat, 121 degrees for 15 minutes. If you're doing that same temperature with dry heat, 600 minutes. That's 10 hours. So 10 hours at the same temperature, dry, or 15 minutes at that temperature, moist. See the difference? Big difference there. It's going to significantly impact your ability to keep things sterile, maintained. Not everything can withstand the moisture though, right? If it rusts or some things might melt because of the heat. So not everything can be autoclaved in order to sterilize it. But where you can autoclave, you can add in moisture, pressure, and heat. Sterilization can be achieved within 15 minutes at that 121 degrees Celsius. Remember, 100 is boiling. All right, bacterial endospores, again, the most resistant that we know of. It's the most resistant microbes of all of them. So usually if you're going to destroy endospores, you're going to need temperatures above boiling. Hint, hint, endospores survive boiling. Boiling doesn't sterilize. Boiling doesn't sterilize. Boiling does not sterilize. I can't say it enough. All right. So if you're boiling something, even for 30 minutes, boiling isn't going to sterilize shit. Endospores are going to survive it. So when you are using, like you're brewing or something like that, it's usually a common problem in brewing. People think that they can just boil their equipment and that would sterilize things. Guess what's going to survive the boiling? Clostridium botulinum. So if you aren't actually sterilizing your equipment, Clostridium could be growing in your fermentation. If your organisms are making acid as a byproduct of fermentation, Clostridium doesn't do very well in acidic environments. So usually high levels of acid or low pH, right? Same thing. Low pHs are friendly for brewers because it helps prevent the growth of Clostridium. But if you're not brewing, if you're doing other things, then you want to be sure Clostridium organisms like Clostridium botulinum or Clostridium tetanum or Clostridium difficile, C. diff, if you're trying to eliminate these things, boiling isn't going to do it. It's not going to get rid of C. diff. You know, so we're already talking about eliminating endospores, and I'm already telling you ways that endospores can't be eliminated. You have to autoclave it, basically. Washing your hands and using the typical things that we use to clean surfaces like Lysol and whatever, they don't get rid of endospores.
It's like 99.9.
99.9, not 100. Okay. Let's mute that. All right. So, who one's at my house? Who knows who? I don't have any idea. Oh, right. They're working on my RO water today. I'm really excited about it. Our RO water has not been working. And we've been on city water for the past couple of weeks, and I can't wait to get my purified water back. I can smell it, you guys. I can smell it in the tap. I can smell the tap water.
I love being a well water.
Yeah. So I'll say that wells have pluses and minuses to them.
It does.
So people say that, but then come taste my RO water.
No, ew.
Why?
Well water tastes
So
better.
well water has minerals in it, right? But well water's not controlled by the city, so you can still have microbes in it too. It's not treated. And microbes. So I'm just saying, I wouldn't be a well water drinker if I had the choice, and this is coming from a microbiologist. So the minerals that are in it can be good for you, but it just depends on the well, right? If you're having it tested and you're treating it properly, then your well water can be safe. But just having a well and having well water, that's what led to cholera, right? In London. I'm just saying.
Thankfully, my mom was a wastewater technician.
Yes, that helps, right?
You
So
know,
being aware of
you
that
do.
stuff, being aware of water quality. Anyways, moving on to this. Right, so thermal death. So you have to have it hot enough for long enough in order to kill the endospores. That's the point here, right? And each organism, its vegetative form even varies. So some of these vegetative guys, even though they're not endospores, some of these vegetative guys are more hardy than others. So the whole idea is whatever temperature you're at and for whatever time you're doing it for, you need to be sure you're doing it long enough to get rid of the microbes you're trying to get rid of. If you're trying to sterilize, boiling ain't gonna cut it. Okay. So susceptibility of microbes to heat. We have these two terms, thermal death time and thermal death point. Thermal death time, the length of time that it takes to kill all microbes at a specific temperature. So if we're talking about 110 degrees Celsius, how long does it take at 110 degrees Celsius to get rid of all the microbes? That's the thermal death time. The thermal death point is temperature. So it's a point. It's like a temperature point, okay? It's the temperature required to kill all microbes in 10 minutes. So if you had 10 minutes, what temperature do we need to go at to kill all microbes? That's the thermal death. Yep. So it's a thermal death point, the lowest temperature that you would need to kill all the microbes in that situation in 10 minutes. All right. You understand the difference?
Mm-hmm.
So it's looking at exposure time versus the temperature in a set time. All right, so like we said, boiling water. Boiling water is for disinfection, not for sterilization. All right. So you could have a boiling water bath or a chamber in order to disinfect things. This is perfectly useful for treating things, for disinfection of contaminated, linens, or, if you have somebody in your home that had COVID, boiling will get rid of COVID. Okay, so if you had somebody that was having a viral infection in your home, you could boil like their cups and their laundry and things like that so that you wouldn't risk exposure to other family members. Boiling would be perfectly fine for stuff like that. Boiling will get rid of lice, yes. So the idea is that this is pretty effective. You can do it at home. It's not going to sanitize, and it's pretty easy to recontaminate the thing. Just once you've done boiling it and it's dried and you reuse the thing, it's contaminated immediately. So it doesn't prevent contamination, but it can remove and disinfect pretty well. It's pretty effective for that. We also know pasteurization is probably something you've heard, that your milk is pasteurized. This is also just disinfection. Duh. Like, if you didn't know that your milk isn't sterile, I mean, what do you think happens? Why do you think it goes bad in a week in your fridge? There's microbes in it. It's not sterile, right? Microbes do not spontaneously generate. We've already learned about that. So the reason that your food goes bad in your fridge is because there's microbes in it. So we have a couple of processes that you can go through with pasteurization. There's pasteurization and there's ultra pasteurization. So pasteurization, again, and ultra pasteurization, just a matter of how high the temperature is and how long the exposure is, and if you're filtering the beverage afterwards and things like that. So pasteurization is literally like bringing the temperature up to a sub-boiling temperature. We're talking like up here, it says 71.6 degrees for 15 seconds, or it could be 63 to 66 degrees Celsius for 30 minutes. Those temperatures, 71 to 66, not a big difference there. We're talking like 5 or 6 degrees, right? And it's 15 seconds versus 30 minutes. That's the difference already, you can see. So you could expose it for the lower temperature for a longer time or the higher temperature for a shorter time. But either way, it's not this one time that you're going to do it. You're going to cycle it. So you will hit it for 15 seconds at 71 degrees and then cool it down, and then hit it again, and then repeat this repeatedly until you have eliminated enough pathogens to your satisfaction, or according to what the FDA's regulations are for milk, for example. So you're allowed to have microbes in milk. You're not allowed to have any actual true pathogens in the milk. And then the milk will go bad over time because we haven't sterilized the milk. There's still organisms in it. Yes.
Why does Brahms milk stay fresher so much longer than any other brand? Is it just 'cause
Probably-
it's
We get it
local?
from locally, does have to ship here.
Why do you think that being locally would make a
Does
difference?
have to ship. It's
Because.
But what about Highland?
The shelf life.
Highland is made in Oklahoma. Yeah, right. I agree. I like Highland milk. So I like Brahms. I haven't looked at their pasteurization process, but it might have to do with pasteurization. So does anybody here buy lactose-free milk,
The A2.
like Lactaid or anything like that? Okay, I recommend it. You don't drink a lot of milk. If you're in a family that drinks a lot of milk and you're going through your milk and you're not throwing it down the sink, you're fine. Do whatever you want to do. But if you aren't drinking a lot of milk and you like milk occasionally, like with your Oreos or with your cereal just sometimes, or like in your coffee or something, you're not going through it, and you find that you waste milk if you buy actual just milk. Buy Lactaid. It tastes the exact same. Promise. I promise. It tastes the exact same, and it has months of shelf life. Why? Because it's ultra pasteurized. It's treated with more heat to kill more microbes.
It tastes better in macaroni. Almond milk is not good macaroni.
No, almond milk is not good in macaroni. So if you're doing things that you want the milk taste to taste like milk, Lactaid, it tastes like milk. Even the whole milk Lactaid, it just doesn't have lactose in it. It's been treated so that the lactose in it has been digested out
So it's still milk, but without that.
without lactose in it.
But that's the same thing with that A2 milk from Brahms, because they have special cow.
Yeah, I don't know enough about that.
protein.
Yeah.
And Brahms is like the only
It's all
person, like,
pasteurized.
only
Yeah.
company
It's better
that
for
only do
people
type
with
of cows.
lactose.
Especially bread.
The dairy factor in total. Oh. What's
See
because-
that, I don't know where you guys get that idea. They would like, that difference in time, like being transferred just because it's over in Tuttle, like, you guys know that the Highland farm is like just as close. Like, I'm not kidding.
Brahms is still better. I
Like,
don't
it's
know.
on the way to, I'm pretty sure Highland is on the way to Tulsa, I'm pretty sure. Or it's on the way to Tulsa, or it's on the way to Dallas. It's one of the two. It's on the highway. Yeah, Highland is.
No, it's ultra pasteurized and things
It's
too.
because, I'm pretty sure, and I haven't looked, but probably Brahms ultra pasteurized, like not just pasteurized.
That's why we get
It
it.
says they go further than the recommended standards.
Is that what it says on, did you look it up?
Yeah.
Yeah. There you go, ultra pasteurized.
So I'm not crazy.
Or
it's
lactose
just that-
intolerant.
Well, it lasts longer, right? And so that's the difference is like milk, the reason it starts to taste a little off, even before it's bad, right? It gets I hate milk, you guys. I hate it. So, I hate it. I did not grow up in a family that drank milk. And then my first husband was a family that drank milk. Like they would buy gallons of it and freeze
Red
them.
flag.
They had a family of like 7 kids and
That's
so they're
weird.
Catholic and drank a ton of milk. I hate milk. So it's the taste. But what you'll notice if you buy Lactaid, it doesn't have that off-putting taste because it's been ultra pasteurized. And that's probably why Brahms tastes that way. They have that too.
Yeah, they're.
So what they're being fed and how they're being treated, that milk is gonna be,
Genetically modified in cows to not produce that protein.
yeah.
Yeah.
So makes it last longer,
Well,
to
it
not
doesn't
have
have something that's harder for you to digest.
digest, yeah. So I'll say that is like most people, you'd be surprised, actually, a lot of people, this is not me trying to make any comments about certain races, but a lot of people think of the Black population in America as having issues with digesting lactose. It's actually more common in the white population to not be able to digest lactose. It's just, you just don't know that you're not able to digest lactose. And you haven't most people have correlated digestive issues with having drank milk because you would just drink it so much or have cheese so much or whatever that people just don't realize that digestive issues might be related to being lactose intolerant. I'm just saying. Most people just don't know they're lactose intolerant, like dead serious. Like it's extremely common to be lactose intolerant. It's really common. So think about that. Think about buying Lactaid the next time you're at the store. Because when I buy milk, I buy Lactaid. And I swear to God, like that milk will, like it'll say like it lasts for like 3 months on the label. And I've never had it go bad before the sell by date. Even after I open it, it'll sit in there open the whole time. Yes.
What about like Fairlife milk? Is it like every time I bought it, it's good for like 40?
They probably ultra pasteurized. Yeah, I bet that they do. So you can buy ultra pasteurized milk that doesn't lack lactose. I just buy the Lactaid one because I'm lactose intolerant, so is my husband. And you might be surprised. Try it and see if it helps, if you like it better. I like Oreos with milk. That's the only time I have milk. And so I buy Oreos. If I buy Oreos, I buy milk, and I buy Lactaid. Swear to God, it lasts forever. So anyways, moving on, that's pasteurization. It's cycling between below boiling temperatures and cooling it down. And there is a standard for pasteurization with milk. People think that they think raw milk and the freshest milk is the best.
Yeah.
Pasteurize your milk. Okay. There's a reason why the USDA and FDA are going to recommend that you're supposed to be pasteurizing your milk. So this will inactivate most viruses and destroy vegetative states of bacteria as well. So we'll pasteurize most things that because we have things that's still okay to have the microbes in that they're there for a reason, maybe it helps with your biome and things like that, like beer, like the whole process with beer or kombucha or things like that. Like when you have kombucha, that's been pasteurized, right? So it still has microbes in it though. So anyways, so adding in pressure allows for us to sterilize now. Adding the pressure allows you to rise the heat above boiling. So you can get that 120 degrees Celsius, 121 degrees Celsius for at like 15 PSI, which is the pressure. And then brain moment right now. And then adding in the water helps with the coagulation of the proteins. Okay, so pressure, heat, steam. And that's the autoclave. So here's what an autoclave looks like. You aren't going to get tested over how it exactly functions or anything, but just giving an idea. It has a chamber that inside there will be mechanisms to allow for building of pressure, and we'll literally inject steam into it during the process to help with the sterilization of it. And we have ways to test to see if things have been sterilized or not. There's special tape that we'll put on like glassware and things like that that will change colors if it's been autoclaved correctly. All right, so moving on to the dry methods, we've already learned about incineration in the lab. It's how we keep our loops and our needles sterile. So most of the time, Bunsen burner reaches about 1,870 degrees Celsius at its hottest point. Furnaces or incinerators can operate between 800 to 6,500 degrees Celsius. Here we're talking about incineration of entire bodies, right? So that's what we're talking about with our highest temperature furnaces. So this will ignite and reduce microbes or other cells to ash and gas. Carbon dioxide is the gas that will be released from the ignition of this. There could be other things that are given off as well. It's a pretty common way to keep things sterile. You can also use an oven, even your oven at home, to sterilize things. But we're talking about 150 to 180 degrees Celsius for 2 to 4 hours. So if you're popping on your 350 degrees Fahrenheit oven, leave something in there for 2 to 4 hours, that could sterilize. Okay. My husband made a pizza last night. I don't know what he did, but the whole house filled with smoke, like, oh, before the pizzas were even done. I think he put, they weren't really all that greasy, honestly, but like, he just doesn't clean out the oven either. So he'll have something burn in the oven and he'll be like, oh, something burnt in the oven. And I guess he thinks that it just magically disappears sometime in between. Yeah, but he doesn't ever clean it out. And so it just like compounds every time that he uses the oven. I don't cook that much. Because if you think he's doing a bad job, you should see what happens when I cook things. It's not good. I'm very impatient, so it just ends up being bad. So, but yeah, the whole house was like smoke. You would have thought there was a fire, like an actual like freaking fire in the house. But we had to open all the doors and the windows, and it was so windy outside. It was like all wind was literally rushing through our house. It was like 60 degrees within like seconds. It was just freezing in there, and fire alarms were going off. Yeah. I would say that the wind did help a lot with clearing out the smoke. It did, but it was, yeah. But you can sterilize things with your oven at home. And at some places, things that can't be exposed to moisture, this dry oven exposure is a great way to sterilize things. Cold does not sterilize. It merely just stops or slows down growth. In fact, most things aren't going to be killed by freezing. It's actually how we preserve microbes. So if I wanted to study a microbe and I want to keep my sample of it to work from, I'm going to freeze it. So when you guys freeze your food, it doesn't actually eliminate the microbes. It just puts your microbes in stasis. So the chicken that you're putting away in the freezer, salmonella could potentially survive the freezing. So it's not like you get it out of the freezer and it's like clean because it was frozen and it like froze the cells and killed them. No, no, no. You just put them in cryostasis. That's what you did. So typically freezing temperatures that are very, very low, like -70 to -135, we're talking like liquid nitrogen or like those ultra cold freezers are the best for actually preserving the microbes. Because if you're just freezing them in standard freezers, it's not the best for preserving the microbes because they can get killed some, but don't rely on freezing to kill your microbes is what I'm saying. There's no guarantee that they'll be killed, okay? So, but it's a great way to stop their growth. And so you keep things fresher longer by freezing them. So you don't have microbes growing on there, destroying it constantly. So there are microbes like psychrophiles that actually grow at freezing or sub-freezing temperatures. They live at the temperature. Desiccation means drying things out. We know we can dry things out and preserve them just from that. You can combine freezing and desiccation in a process called lyophilization. So when I buy microbes for the lab, by the way, I buy them in little vials and they come in a powder. That's lyophilized powder. So it's been frozen and dried at the same time in order to do that. If you've ever seen a vial where somebody injects like a liquid into it in order like alcohol or water into it and then mixes it up and then can use that to inject somebody, that powder was lyophilized. So it's drying and freezing at the same time. And it preserves it. All right, so radiation. We can use radiation, which is basically energy that's emitted from atomic activity. So when you have energy put into an atom, it can make your electrons go to higher levels of orbitals, and then they can be ejected from those orbitals with enough energy, and that can cause damage to microbes. And so there's different kinds of radiation, right? So there's going to be UV radiation, which is non-ionizing, and then there's going to be ionizing radiation. So anytime you expose microbes to radiation, this is called irradiation, irradiated. And so there's a couple kinds of radiation, ionizing and non-ionizing, like we said. Non-ionizing, the best example we have is UV radiation. And then other kinds of radiation fall into ionizing. This would be X-rays, gamma rays, things like that, okay? So if you go to Chernobyl and you go to the Elephant Foot where the meltdown happened, that's emitting gamma radiation. That is not like UV radiation. That is ionizing radiation. Okay. So here's how ionizing radiation works. You can put up a barrier in front of most types of ionizing radiation, and there are barriers that this will be able to pass through. The stronger the radiation, the more it'll be able to pass through barriers. So lead vests ain't gonna help you in Chernobyl. That radiation can pass through lead. That's saying something because lead is dense, very dense. But we wear lead vests when we do X-rays. X-rays is also ionizing radiation. So you could use X-rays potentially to kill microbes. The X-rays that cause damage to your DNA that can lead to cancer, which is what we suspect killed Marie Curie. those X-rays, are causing damage to your actual DNA. We're literally exciting the electrons and they're shooting off. And then what they do is go and damage nearby molecules, including your DNA. So break apart your bonds in your DNA. Literally your DNA, your proteins, all the molecules in your body are being broken apart by these excited electrons kind of shooting and reacting with everything. And that's what ionizing radiation does. So some levels of these ionizing radiations can be blocked by certain barriers, but a lot of them can pass through a lot of barriers. So X-rays can pass through walls, but we wear the lead vests, or we have special walls in place that will block the radiation so that you don't get exposed as a healthcare worker constantly doing it. A little bit of exposure to this radiation shouldn't cause too much harm. We can withstand, our body can withstand certain amounts of radiation exposure. But at certain levels, like if you're constantly doing, you're an X-ray technician, you're a radiologist, you're going to want those things put in place because you're going to be constantly put in a situation where you're exposed to X-rays. So you're going to need barriers. That's why they don't stand in the room with you when you're getting your X-ray, right? Because they're going to be doing more than just that one, that day, a week, or month. And how many X-rays did you have to get this year personally? Maybe one or two. And these people are doing it every day, multiple times a day, constantly. So that's what it's going to be like working in the healthcare field. They'll be exposed to these things more commonly. All right, so that's ionizing radiation, that the excited electrons are actually causing your molecules to have effects on them, breaking apart your DNA and things like this. Non-ionizing radiation, we already know that UV radiation can cause cancer. It affects your DNA. So what happens is the T in your DNA, we know how T, it binds with A, right? On the other strand. So what happens is UV radiation causes the T's that are next to each other to bind to each other next to each other instead of the A's across. So whenever your DNA polymerase is splitting your things apart to make new cells, and it comes across that, it can't cause the T's to bind with A's anymore. And so it'll cause mutations in your DNA as a result of that. We have ways to repair this in our cells, but if we don't catch it at a certain point in that process, in your cell process of mitosis and everything like that, your checkpoints, then that can lead to permanent damage. All right, so our ionizing radiation, gamma ray machines, X-ray machines, cathode ray, when you go to get treated for cancer with radiation, it's going to be one of these types of radiation. It's going to be ionizing. Okay. When you go to Chernobyl and you get cancer afterwards, within minutes of being exposed to the elephant's foot, your skin will start to slough off and all this stuff. And we see people who had after effects of the dropping of the atomic bombs and things like that. People that weren't killed immediately later on had radiation sickness. Radiation sickness is because their cells got so damaged from those electrons just shooting through all the molecules in their cells. Their cells got so damaged that they couldn't repair it. They couldn't keep up with it. And the things that are involved in repairing it also got damaged by the radiation. Those aren't like spared. So all of their ability to repair their damage, they can't do it, and cells are damaged. So the cells can just slough off. So have you ever heard about the story of that guy that was exposed to, like, he was a nuclear reactor, and like all of his skin sloughed off, and there was nothing they could do for him. Because you can try to put skin grafts on those people, but it doesn't heal. They don't have the ability to heal anymore. Radiation damaged their ability to heal too. So it can be devastating. Non-ionizing radiation, again, UV rays. These are going to be less excited atoms, leading to our bonds kind of bonding, our DNA molecules binding together instead of across onto the appropriate strand. So UV counts as anywhere from 100 to 400 nanometers. A lot of the time, we're going to sit around somewhere around 260 nanometers for it to be the most effective. So the lamps that we use, like outside of Integris, you notice how the Integris and other places too are now doing it too with the purple lights. When it's like outside, you'll notice that they have purple lights going with UV lights. It's literally UV lights because who's going in to get care at our urgent care clinics? Sick people. So it's to help keep the microbes from spreading in the air from person to person, so that people who are near or around the clinic don't get exposed to that stuff. So it keeps that area a little bit more disinfected. UV radiation can be used for sterilization, but it doesn't penetrate. That's the problem with UV radiation. It can't penetrate surfaces. So it's like only the surface can get sterilized. And you really need exposure of like 30 minutes to an hour at that 254 nanometers in order for it to sterilize the surface. So we don't use it very much for sterilization. Whereas I can take, a food and put it in a package and then put it in like a microwave, not a microwave, but a gamma ray wave kind of apparatus. and then expose it to ionizing radiation. And that entire piece of food, the chicken breast or whatever it is, gets completely irradiated by ionizing radiation. And then it is completely sterile through and through the entire thing. So that is useful. So when you guys see those little packets of like, when you're putting together like for phlebotomy, the little needles, they're in those individual packets, or the little vacutainer accessor thing, those are in little individual packets. Those were put in the packets, and then the whole packet put into an ionizing chamber, and they were irradiated. And so the whole thing is sterilized all the way through that way. It's not like we autoclaved that. We irradiated it. Most of the things that you're exposed to that have been sterilized probably irradiated. Okay. We can also decontaminate through filtration. This is pretty self-explanatory, so I'm not going to go over it. And then osmotic pressure. Again, we've gone over this with hypertonic solutions drawing out liquid. So packing something in salt or high sugar, like we do with preserves, can control microbial growth as well. So we're going to move on to the modes of chemical control next. We have solids, liquids, and gases, but most of the time we operate with things in liquid or sometimes gas form, depending on the situation. So we can fill whole rooms with these gases and then just sterilize everything in the room. So we can do massive chambers. We call them chemiclaves when we do that. So we have the autoclave with the moisture, pressure, and the heat, but a chemiclave, it's like just a chamber filled with gas, chemical. Okay. So aqueous solutions, if something is an aqueous solution, it's dissolved in water, but a tincture is something that has any alcohol added in. It could be all alcohol, or it could just be water alcohol mixture, but if it has any alcohol, it's a tincture. So we have high-level germicides that are going to be used on our critical care items. These are things that have to be sterilized. We will treat things with chemicals if they aren't heat sterilizable, or if radiation isn't effective for them for some reason. Right? We often use high-level germicides on things like catheters, heart, lung equipment, or implants. Our intermediate level germicides are going to be used on things that we consider semi-critical items. Again, things that come into contact with the mucous membranes. So this would include respiratory equipment or thermometers. And then our low-level germicides, this would be areas like tabletops and surfaces and things like that. So these only are going to eliminate our vegetative bacteria and some viruses and some fungal cells. These are not incredibly effective, but they are effective usually for day-to-day use. This would include our electrodes, any straps, furniture that touches skin, but not mucous membranes. Okay. So again, these target the same structures in the cells that the physical methods do. Cell wall, cell membrane, and then DNA, RNA, or protein production. Let's move on to the actual classes. We have our halogens. These are literal halogens on the chemical table. So fluorine, chlorine, bromine, and iodine. The second one over from the right. Those are all of our halogens. So these guys are actually pretty good for sterilization. They can sterilize if they are used appropriately in the appropriate concentrations for the appropriate amount of time. So chlorine, our first example here, these can target and kill endospores. So they can, chlorine products can, if used appropriately, sterilize. So most of the time, whenever you're buying like Lysol or something, it's usually a chlorine-based compound. Could have other chemicals in there as well. So it gets close to sterilization, but it's not at a high enough concentration usually to sterilize, because once we get to that high enough concentration for sterilizing, it's often dangerous for people to be exposed to as well. So that's the problem. But bleach can be used to sterilize at a high enough concentration, for example, a lot of our chlorine-based things. This is why we pump chlorine gas through water when we're treating it in a water treatment plant. So the chlorine will kill all the microbes that we need to sterilize if we're using it appropriately. Okay, so this includes chlorine, hypochlorite. Anybody heard of like hypochlorous acid? Our neutrophils release that. It's a way of fighting off microbes. You can like buy it now in skincare products. So I actually use a spray that has hypochlorous acid in it on my own skin. But these are very effective for controlling microbial growth. They can be in gas or liquid form. Target microbes, again, you can slowly kill endospores, so if it's used appropriately, you could potentially sterilize. But they are very effective. Iodophores, again, another halogen, so iodine. Iodophores are a complex of iodine and alcohol, so we're talking about tinctures here. So we can use iodine on skin. Examples would be like betadine, povidone iodine, and isodyne. If you ever see the brownish liquid that's put on skin, for some people, it's used less frequently now. But it is used in some cases, especially in cases where people are allergic to, what is the name of it? I can't think, my brain's We'll get there. We're coming to it next. So, but you can still use iodine in some cases for prepping things like central lines, prepping for surgery. That brownish liquid that we're using to prep skin, it leaves like a yellow tinge to the skin. That's iodine. Okay. It can be very irritating to tissue, so that's why we have moved away from using it. Next is phenol, chloramphenicol. There it is. Okay, chloramphenicol. So that one is used as a preparation. It's a clear, and it's been favored for making central line prep or IV prep and things like that. Chloramphenicol has, but anyways, it's dangerous. And there's another one that we're going to talk about too. So phenol, anything that's based off of a phenol is called a phenolic, but phenol itself is carbolic acid. It is pretty effective, but it's really irritating to humans and it can be toxic. So we don't like to use phenol by itself, but we'll still use it for decontaminating certain like pet cages or things like in areas in vet clinics and stuff, you can still use it for decontamination. So our phenolics are based off of phenol, and phenol is still considered like the standard, which was used originally by Joseph Lister. It's carbolic acid. So phenolics are based off of that, but these guys are not sterilizing. They're just disinfectants, okay? So chlorhexidine is the other, this is the one that I was really trying to think of. Chlorhexidine is commonly used for prep now, for IVs and blood draws and things like that. Go donate blood. They'll sit there and rub your arm for 30 seconds with chlorhexidine, but people can get allergic to that. They can get allergic to that, and you can be allergic to iodine. If you're allergic to both chlorhexidine and iodine, you cannot donate blood, period. Because they'll use chlorhexidine first. If you're allergic to that, they'll move to iodine, but if you're allergic to both, they can't draw your blood. It's just, they can't keep it sterile enough. They can't keep it disinfected enough, really, in order for it to be safe and poke a hole in your body the size of those needles. When you go to donate blood, you are using a needle that's 16 gauge. That's 16 on the inside. So overall, the outside is usually about 14 gauge. It's a pretty big needle. So, yeah, I don't know. Like my nose ring is like 16 gauge. It's not 14. The smaller the number, the bigger the gauge. So to put that in perspective, most earring holes are 20 gauge, 20 to 22 gauge. But yes, so butterfly needles are 22 typically. 20 or 22, depending on what brand you're buying. So those are the tiniest needles you can get, but anytime you're using a super tiny needle like that, you're risking breaking open your red blood cells. And so when you donate blood, they can't use the tiny needles because it will break open your red blood cells. And that's what they're trying to get from you is your red blood cells. So they can't risk hemolysis. One of the biggest issues I can tell you if you're going into nursing or into vet care and you're going to be drawing blood, that if the lab calls you and tells you that your sample is hemolyzed, I cannot tell you how many times I will call down to the floor and be like, okay, we need you to redraw the sample because it's hemolyzed. And then being like, then what did you do with it? Because I didn't hemolyze it. You hemolyzed it. The lab can't. There's nothing the lab does that can hemolyze it. So hemolysis, breaking open the red blood cells. All the lab does is take your tube, let it sit there for a while, and then spin it in a centrifuge. They're treating everybody's sample that same way. They didn't break open your red blood cells by centrifuging it. When nobody else's red blood cells are breaking up, and it's impossible to do that. So I'm just saying, if the lab calls you and tells you that your sample is hemolyzed, it's probably because you're using a butterfly needle in an area that has been poked several times and already has bruising in that area. And so you're just drawing in the bruised, damaged blood cells from that area a lot of times. You'll almost likely have to stick them somewhere else. Just saying. And so there are some tests that you will need to do as a nurse that you have to have a certain amount of blood in that tube in order for it to work. And it cannot be hemolyzed. You're going to get calls repeatedly about how your samples have failed to meet those qualifications. Those qualifications are there for a reason. It just, the test depends on those things to meet those things. It's not the lab being mean to you. They are not picking on. Believe me, they don't want to call you. They don't want to make you redraw. So just keep that in mind when you're doing your blood draws, because as nurses, you're going to be doing a lot of blood draws. If you work at Mercy, you might be lucky. You might have phlebotomists that work on the floor for you. But Integris doesn't. Their nurses do most of their blood draws for them. ER is a little bit different, but not on the floor. If you're on the floor, you're doing your own blood draws. All right. Moving on. Chlorhexidine can also be added to mouthwashes and body washes. If you've ever heard of Hibiclens. Hibiclens is used to remove microbes from like the body, as well as used in surgical scrubs. It's literally what they use in surgical scrubs. It's that the soap that's in the soap to sterilize those well, to disinfect those surfaces, okay? Alcohol also does not sterilize. It's disinfectant, okay? There's no concentration of alcohol that's gonna sterilize. Now, we're gonna talk about the concentrations of these guys. Concentrations of 50% and higher of alcohol will dissolve membrane lipids, disrupt cell surface tension, and compromise the membrane integrity. So if you want to affect the membranes, the alcohol is good. The alcohol also affects the cell walls of these guys, which we mentioned earlier. But alcohol can affect cell walls and cell membranes. And if you have water in your alcohol, you can also coagulate proteins at the same time. So these guys are actually really effective. They're just not going to sterilize. They're very effective. In fact, alcohols work the best at a 70% concentration. So the next time that you are at CVS, find some isopropyl alcohol or ethyl alcohol for use around home for cleaning wounds. Don't buy the 95%, buy the 70. It works better. Science says 70%. The water that's there, that 30% water, helps with the coagulation of proteins. That makes it more effective. That's why all of our hand sanitizers are 70%, not 90. Not because it's expensive to have 90, but because 70% actually works better. All right. Isopropyl alcohol is not only cheaper than ethyl alcohol, but it works better than ethyl alcohol. The reason that we use ethyl alcohol a lot of times, I'm sure you guys have noticed, like this is ethyl alcohol. All of our hand sanitizers have moved to using ethyl alcohol instead of isopropyl alcohol. Does anybody know why that is? It's better for your skin. So the isopropyl alcohol is so effective that it'll dry out your skin more. And because after COVID, we had people using hand sanitizer. We didn't want to basically cause damage. I hate the way alcohol smells when it dries off your hands though. Okay. So anyways, as the ethyl alcohol dries off your hand, it's less likely to cause damage to your barriers, your skin barriers. And so with isopropyl alcohol, the skin barrier stuff can actually cause enough damage to your skin barrier to cause microbes to be able to get into your body easier. So they switched to ethyl alcohol in order to move towards maintaining your skin barrier integrity. So we knew you were using it so much after COVID. Make sense? All right, moving on. Oxidizing agents, anything that has oxygen in it as an active ingredient, basically. And this includes hydrogen peroxide. Okay, so hydrogen peroxide, its chemical formula is H2O2. This is a biggie here, H2O2. So peracetic acid also counts here, but the one we'll be focusing on for the exam and everything like that will be hydrogen peroxide. Okay, so these guys form oxygen-based free radicals. These are highly toxic to cells, especially the guys that are anaerobes. So these can be sporicidal in high concentration. So when you guys go to buy hydrogen peroxide at the store in that brown little bottle, it's at 3%, and it's not a high concentration. That's not going to be sporicidal. So that's going to be a disinfectant, not a sterilizer. But you can sterilize with high concentrations. If you get concentrations of 35% or higher of hydrogen peroxide, you can sterilize. And you can actually use concentrated hydrogen peroxide in basically a gaseous form in order to sterilize like a room or surface or things like that. So you can use that as a gas in order to sterilize in a chemical cabinet. So it is colorless. It's has a slight smell to it, but it's not super astringent or anything. It is caustic because it has the oxygen reactive aspects to it. It's toxic because of its oxygen, and it can be bactericidal, fungicidal, virucidal, and sporicidal if you use it at high concentrations. Like we said, up to like 35% is sterilizing concentration. And so we can vaporize the hydrogen peroxide and use it to sterilize equipment and things like that. Moving on to our detergents. Detergents, again, remember how we talked about surfactants before? And I said they had a charged head and a long tail. Well, surfactants are detergents. Okay. So a detergent they're again polar molecules that act as surfactants. So the surfactant is the action that it has. The detergent is the name of like what kind of chemical compound it is. Okay, they're the same. Think of them as interchangeable for our purposes, right? So again, these are going to interact with our membrane. They've got the charged head. In this case, we're giving an example of a positive head and then a long non-polar tail. All right, so this would be the hydrocarbon tail here. Or we could have our head be this whole big old area here, and then this would be your tail on this CnH And basically numbers of carbons and hydrogens, that'll be your tail on that one, okay? So benzalkonium chloride, you probably heard of this one and you probably used it. Anybody that went and got their ears pierced at like a Claire's or something like that, they usually give you a solution to clean your ear piercing, and that's usually benzalkonium chloride. Benzalkonium chloride is okay for using it as an antiseptic, it's pretty safe for use on skin, but it's a surfactant. It helps break down the integrity of cell membranes. That's what that does. And so it works the same way as our detergents. These again, our surfactants are not used for sterilization. They are used for disinfection, and they're often combined with other chemicals. They'll break down the integrity of the membrane and allow other chemicals to get inside or cause the cell to leak out stuff. So quaternary ammonia compounds. A quaternary ammonia compound is like this one up here. This is a quaternary ammonia compound. It has a nitrogen, and NH4. That's a 4. I promise it wasn't just a 2. NH4. This is ammonia. And so anytime you have N surrounded by crap, that's a quaternary ammonia compound. So it's a nitrogen with 4 things around it. Okay, so it's a quaternary ammonia compound. So we call them quats. So our quats are our surfactants or detergents, same thing for us, that are great for their surfactant properties, but usually aren't that effective at just sanitizing on their own. And so we often combine them with other things. They're weak microbicides, okay? And then things like Pseudomonas, which is a very, very common bacteria that's on like almost all surfaces that we're exposed to. It grows even abundantly, even in soaps, and it can actually use aspects of the soap in order to live. So our soap trays where you're having your little soap dish sitting out and it starts to look gunky or nasty to you, that's probably because Pseudomonas is growing on it. Our heavy metal compounds, these guys can basically inhibit the function of our proteins. That could be through competitive or non-competitive inhibition. You can become allergic to these metals as well, but mercury, silver, and gold are often the ones that are most commonly used, heavy metals, in order to treat things. We know silver can be incorporated into a lot of things. You can buy scrubs now that have fabrics with silver basically woven into it. You can buy countertops that have silver embedded in them, and this is all in order to have antimicrobial activity. Aldehydes. So we have heard probably of formaldehyde and glutaraldehyde and all these things. They have this functional group on them, CHO functional group, that's an aldehyde group. So glutaraldehyde, it can be, so these aldehydes in general can potentially kill endospores, right? So these can potentially be sterilizing agents. In case you haven't noticed though, they're pretty unfriendly to living organisms. So formaldehyde and glutaraldehyde, you typically aren't going to use this on living organisms, right? We'll use this for embalming fluids to kill the microbes that are inside of the dead bodies so that we can, show them for viewing at funerals and things like that. So glutaraldehyde is great. It can sterilize. Formaldehyde, we also have formalin, which is the aqueous solution of formaldehyde. It's mixed with water. This is an intermediate to high level disinfectant. If you're using it at high enough concentrations, it can sterilize, but we typically use it at lower concentrations. We use this for embalming. And then there's another one called orthophthalhaldehyde. You're welcome that I don't make you spell this word. It took me a long time just to be able to say it. It's also abbreviated as OPA. It does not sterilize. So it doesn't destroy endospores, but it's a lot less toxic and it's a lot more user-friendly for people. Okay, so it's also more expensive. Next, we're gonna move on to our gases. The major gas that I need you to be aware of is ethylene oxide. This is basically, so ethylene oxide is basically used as a gas in order to sterilize like large areas. So you can do big equipment, basically sitting in a room in an area that we will call a chemiclave. So this reacts with DNA and proteins and inhibits their ability to act and function. Chlorine dioxide is another one that we can use for gas sterilization. We use chlorine dioxide for treating drinking water and stuff like that. The problem with ethylene oxide, and the reason we can't use it just like all the time on everything, is because it's explosive and it's carcinogenic. So if it affects DNA, and that's how it affects the microbes, it affects your DNA too. So it's carcinogenic and explosive. So when you are using this in order to treat areas, you would need to have a proper ventilation system. And you're usually going to treat it on its way out of the ventilation system as well. So it's not going to affect, the life nearby. So they used ethylene oxide in order to treat, was that the Senate or the House of Representatives? I think it was the House of Representatives, was it the Senate? I can't remember. Oh, does it? Yes, there it is. Okay, yeah, it is there. So they use chlorine dioxide, that's my fault. They use one of these gases, but they use chlorine dioxide in order to disinfect Senate after they were attacked with anthrax. Literally, they mailed them mail with anthrax powder in it. And when it got exposed to the Senate offices, they had to disinfect the entire offices. So they use chlorine dioxide in order to do that. Acids and alkalis, we already know that changing the pH affects your hydrogen bonds in your proteins and your DNA and things like that. So changing your pH by using alkaline or acidic solutions can be used. These are not going to sterilize typically. These are just going to be able to cause disinfection. So this could include our alkaline one, our ammonium compounds, and we also have our organic acids. This includes acetic acid, propionic acid, lactic acid, benzoic acid, and sorbic acid. That we can also use these as preservatives in food because these are generally pretty safe for exposure to people. And things like ascorbic acid could be based off of things like vitamin C, and this is just vitamins that we need to be exposed to anyways. Acetic acid is vinegar. So it's like the acid that makes vinegar vinegar. So when people are telling you to clean your houses with vinegar, they're talking about using an acid solution in order to clean, disinfect. All right, so here's some examples of some of the chemicals that we're associated with. So we have our questions here. I didn't write the names on the board, so you're going to get called out. Get ready. So our first question is, which microbes are most resistant to control methods? This is going to be number 2. I want to get it pulled up. Notes. Number 2. Please stop emailing me while I'm trying to open my notes. Number 2 is Emily Atolano. Yes. So endospores are definitely our most resistant guys. All right, so 26 is me. So why is moist heat more effective at sterilization than dry heat? Because the moisture in there helps with the coagulation of the proteins and the destabilization of them. All right, so our third question is number 23. This will be Levi. Levi, give an example of a halogen control agent. Great example. We also have chlorine. People use like bromine in like their hot tubs and things like that. So yeah. All right, our fourth question is number 21. This is going to be AJ. AJ, why are alcohols more effective at 70% instead of at like 100%?
I'm not sure.
You're fine. So we just talked about it and I didn't give you a heads up. Yes.
So sorry, may I answer first?
Yeah, of course.
Because unlike 100%, they will not dehydrate what they absorb.
Right, so the water actually helps with the coagulation of the proteins. So interacting with the actual water molecules helps the proteins to stick together so they're less likely to function with their normal functions. So coagulation of proteins. And then our last question is number 5. This is Lily. How do surfactants work? Just basic explanation. Yeah, exactly. They mess up our phospholipid layer of our membranes. And that is it for today. We'll go up to the lab. We've got the ELISA lab. So fair warning to you guys, because you remember on our lab, okay, we got a 50%. Okay, the class after you guys got a 44. So from now on, from your first lower than 50 on to that lower than 50, they're counting for points. Okay, so keep it above 50 and we'll keep it at participation score. But if I see anybody in this section, once this section drops below 50% average, that day onward, points. Okay, and that's going to be for each section. Yes. So it's based on the questions that you completed. So if you only got one question in and you got it wrong, you get 0 points. So if it gives you, like, if you completed, because you'll go through like the ton questions sometimes multiple times, or you might only go through 3 questions. Whatever your percentage is on Kahoot, that's going to be your points out of 10. Yes.
So my question is about the lab exam, and you might have said this, but I have to hear things a couple times for it to sink in. It's 50 questions. Are all of them going to be us looking in the microscope and answering those questions, or there going to be like
There's like
multiple choice, true, false, and then some we have to look in the microscope and identify the test?
Right, so there's 50 questions. They're all multiple choice. Okay, and then there will be about 10. I want to say there's 9 or 10 microscope associated. Well, there's might be more than one question associated with each microscope. There will be like 9 microscope stations. Okay, you might have 1 or 2 questions from that microscope station. Okay. There's like one section that'll have 3 questions, but I promise that one of them should be like super easy. There's multiple choices, like, so there's like, there will be no more than 5 choices on the multiple choice either. So it's normal multiple choice setup, okay? You do not have to find the microbe in the microscope either. It will be focused for you. If you get to a microscope, you are welcome to use define adjustment, but if you can't get it focused on define adjustment, you can call me over. It does not cost you any points to call me over to fix the microscope and focus it for you. Okay? Huh?
We have computers for.
Yes, it's on your computers. It's given just like your lab, your courses, or your lab, your lecture exams. It's on Canvas. It's the normal Canvas stuff.
Yeah, that's a good question.
Yes.
Sure,
Yeah, you can freeze milk. Sorry, Haley. Yeah.
What?
It doesn't taste the same. does
No, it doesn't.
it?
Surfactants, how they actually.
The membranes.
The membranes and everything. How is that different than pulmonary surfactant that holds open our alveoli?
So that the whole idea with the pulmonary surfactants and how they work. So I don't know enough about the actual chemical like makeup of those, but the idea is to basically break open like the mucus and stuff like that. So it doesn't just like affect the membranes, right? And it's made at a certain concentration and compatibility with the cells that are in your lungs so that it can help break down the mucus and therefore hopefully the alveoli won't be clogged up and we can get exposed to the air that way. So there's literally surfactants that are designed for breaking down the fluid in the lungs
So there's
as
just different
opposed to,
compositions
yeah.
of them,
Mhm.
but they're all
Yeah.
empathic.
So they're all antipathic. And that's just the idea of how they work with whatever molecule they're targeting.
Okay.
So you're just thinking at the higher level than what I expected everybody else to be thinking at, which is like, oh, surfactants, they interact with the membrane, but surfactants can interact with other molecules.
It's kind of hard
as
going
well.
through, because this is technically an intro to micro, right?
Yeah.
It's kind of hard going back through these, because they weren't required to me going through like the paramedic and having to learn all the medicine
Mhm.
and the A&P and patho and stuff.
Right, and then
So-
learning the application of the
Yeah.
thing, and then the other side of it that you didn't have to. Yeah.
Right. So now I'm going through like nursing school right now.
And trying to make sense of it.
Right, all that.
There's advantages and
Cross
dis,
my head.
right, it's the advantages and dis